Hello guys, welcome back to my YouTube channel Dr. R.kmedicos. In this video we are going to discussing about morphology of bacteria and anatomy of bacteria. So continue to watch my video at last and never escape this video. And if you are new at my YouTube channel, then please subscribe, like and share my YouTube channel and never forget to press the bell icon so that you will get notification of all video uploaded by me on my YouTube channel. Let's start this video without escaping the time. In this video, firstly we will discuss about the classification of bacteria on the basis of gram strain. As you already know that on the basis of gram strain, the bacteria are classified into two broad categories, and first is gram positive bacteria, and the gram positive bacteria are those that resist decolorization and retain the primary stain appear violet. And now the gram negative bacteria. Gram-negative bacteria are decolorized by organic solvent and therefore take the counter stain appearing red. Now we will discuss about the mechanism of gram staining. And the exact mechanism of gram staining is not understood at now. But the some mechanism are based on some assumptions and which are following. And first is the gram positive cells have more acidic protoplasm, which may account for their retaining the basic primary dye and more strongly than the gram negative bacteria. And in case of gram negative bacteria, the gram reaction may be related to the permeability of bacterial cell wall and the cytoplasmic membrane to the dye-iodine complex, but not the gram positive cells, as you already know. And the gram positive bacteria become gram negative when their cell wall damaged, because the mechanism of gram staining according to some assumptions based on the permeability of bacterial cell wall. So the gram positive bacteria become gram negative when the cell wall is get damaged. Now we will discuss about the mycoplasma. As you already know that the mycoplasma is a bacteria that lack the cell wall, so that they are differ not merely in staining characteristic but also do not possess a stable structure and several other properties and morphology. And they occur as a round or oval body, such as growth requirement, susceptibility to, and as a interlacing filament. Now we are going to discussing about the acid fast stain. As you already know that the acid fast stain was discovered by Ehrlich, and he found that after staining with aniline dye, penicillin, bacteria lost their distinctive shape. This method, and that is the acid fast staining method modified by Nielsen. Nowadays this method is adversely used by the scientist for staining of bacteria. In this method, the cocci on the application of heat, it get decolorized with maybe arranged in pair, that is also known as diplococci, or maybe arranged in chain, and the 20% sulfuric acid and the counter stained with the group of 4, that is the tetrad or 8 a contrasting dye such as methylene blue. The acid fast bacteria such as Streptophilococci retain the fuchsine, that is the red color pigment, while some Bacilli too may be arranged in the chain, the other take the counter stain. The acid fastness is not a property division of lipid alone, but also depends upon the integrity of cell wall. Now we will discuss about the classification of bacteria on the basis of their shape. And on the basis of shape, the bacteria are mainly classified into 6 categories. And first is Cocci, and which are spherical or oval in shape. And the second is Bacilli, and that are rod-shaped cell, and Vibrios, and that are comma-shaped, and the Aspidilla are rigid spiral form, and Aspidochetes, and that are flaccous spiral form, well as the actinomycetes show the branching filamentous form, right? These are the classification of bacteria on the basis of their shape. Now we will discuss about the bacterial anatomy. The outer layer of or that is also known as cell envelope consists of different species, right? In general, the wall of gram-positive bacteria having two components, a rigid cell wall, and beneath it there is the other cell envelope which have the similar chemical nature than the cytoplasmic or the plasma membrane and the envelope those of gram-negative bacteria, right? And the lipopolysaccharide present on the beside of the cell wall, and the cell may be their endotoxic activity and antigen specificity enclosed in a viscid layer and which may be a loose sheath, and they were formerly known as bovine antigen, right? In some bacteria, the lipopolysaccharide consists of the three region, and we will discuss region one by one region in detail. And region first is the Quiery filamentous appendage protruding from the polysaccharide portion, determining the antigen cell surface, the flagella, which are the organ of specificity. Region second is the core of polysaccharide and which help in locomotion, and the third region is the glycolipid portion and the organ for the adhesion and is responsible for the endotoxic activity. These are the all about the three region of lipopolysaccharide which are present in some bacteria, not in all bacteria, right? Now we'll discuss about the second layer of cell envelope, that is cell wall. The cell wall responsible for the pyrogenicity, lethal effect, tissue necrosis, shape of the bacterial cell, and the confer on it rigidity, anti-complexity activity, and B cell mutagenicity and ductility. And these all are due to the cell wall. And the cell wall cannot be seen by the direct improvement property and anti-tumor activity. It may be demonstrated by the plasmolysis. In the plasmolysis, cell wall is called the outer membrane, which, when placed in hypertonic solution, the contains various protein known as outer membrane cytoplasm loses water by the osmosis and shrink, and this process is known as plasmolysis. The cell wall contain porine protein, retain its original shape and size, and form the transmembrane pores that serves as a diffusion, and that is also known as bacterial host. And the cell wall may also be channel for the small molecule. And now we will discuss about the innovation of cell wall synthesis. Cell wall synthesis may be innovated by many differential staining procedure or by the electron factor. Chemically, the cell lysosome acts on the gram-positive bacterium in a wall is composed of mucopolysaccharide, that is also known as mucopeptide, and peptidoglycan hypertonic solution. A protoplasm is formed, and that is also known as murine. A scaffolding formed by an acetyl consisting of the cytoplasmic membrane and its glucosamine and N-acetylmuramic acid molecules contain, right? This is all about the cell envelope, and the remain part of cell envelope we will discuss about this in next video. So please subscribe, like my YouTube channel to get the latest update about incoming video. Thank you for watching my video at last.